Bring in one blue Scantron. Don't fold it up. They don't go through the scanners. They get kicked out of the scanners. And also bring in a couple of sharpened pencils, the number 2 pencils. These are just dumb machines. All they read is the marks on the machine. All I'm going to want on that will be your name, your Z number, and the exam number. So that'll all be printed at the top. We'll do that, I think, the first time you do it. Not a problem. Just come in, get settled down like you are now, put everything away, then we'll start handing them out. The exam will have to end promptly at 3:30 because we're going to probably have a whole stack of exams up here, probably 4 or 5 grad students in here helping out, keeping all of this proper. At the request of a few of you or something, I did add an extra page to the back of the exam that you can use as scratch paper. You're not to remove that. Everything has to be returned. So you can write on the exam. Again, we won't answer any questions about the exam. If you think there's 2 correct answers or no correct answer or whatever, just make a notation. I'll have your exam available, the actual printed copy. You can come to my office when you want to review the exam, and we can discuss it at that point. Another quick point, I don't know whether I should go in this direction or not. I mean, there's always, raise your hand if you have copies of my old exams. Okay. There's always these rumors out there that there's a full set of my exams out there, and for God's sakes, please don't pay for them. Okay? There is one exam that I know of. I think it's actually exam 1. You can find it as well as I can with a Google search. It has a question about chi-square, so I know it goes back at least 5 or 6 years, something like that. Now, you can gamble. You can think that I'm just getting old and frail and lazy, and I've used these same questions year after year without changing anything. If you're that kind of a risky investor, I've got some derivatives I'd like to sell you. I had a student, I don't know what, I'll try this quick. Last year, every couple of years, there's always a student who comes into my office feeling compelled to tell me they're going to get an A in my class. They're going to do this. So I remembered her. And then what I noticed was when I got her exam back, her score was so low that I thought that I probably got it in with the wrong version. Usually as careful as we try to be with this many students, 1 or 2 scantrons get put in with the wrong answer key, and I got to go back and rescore them by hand. And this was such a ridiculously low score that I figured this is what happened. No, it was in fact the right version. Then I got a little bit suspicious, and I started checking, and lo and behold, she had used the exact answer that had been on that exam posted on the web. Did a wonderful job of memorizing the answers to all those questions, and then failed the exam, and I never saw her again. So I've given you example questions. If they're out there, I've seen a couple of other ones. They're riddled with mistakes. Don't buy into it. If you're a gambler, do it. But I keep rewriting these. I keep putting in new questions. Just because it looks like a question that even I gave you, read carefully. Okay, that's probably the single best piece of advice I can give you. So many students just grumble when they come and review their exam when they realize they knew the answer, they just didn't read carefully. Okay. Yes?
Is the official policy that we are not to look at exams?
Yes, it's academic dishonesty. If you get to them, I'm just saying don't bet your grade on them. You'll be unhappy. Now if somebody finds some, I'd be interested in seeing them. I just would like to see what is out there. But as I said, please don't pay for them. All right, so we got that. So what we're going to do today is we're going to finish chapter 6, which I think is for the exam 2. It's going to be probably the shortest, least amount of material, and then we're going to start into chapter 7 on genetic recombination. That's a pretty intense, fairly long chapter. It's going to take at least 2 lectures to get through, and please don't tune me out just because it's going to be on exam 2. I think you're going to really, for a lot of you, it'll make more sense if I at least walk you through some of this. We're going to start moving more into conceptually and problem-solving type basis of genetics. So and then at the end, I'll stop probably around 25 after 3, and then I'll just open up for a discussion of anything related to the exam. At that time, I'll start this in-class registration for the iClicker. So I think the way it should be set up, if your name doesn't come up, then it means your iClicker is registered. What you'll see is a group of about 5 names. It's just last name, first name, and then there's a letter next to it. You see your name, you press the first letter, a second letter comes up, you hit the second letter, you're done. Below it, you'll see the next 5 names that are coming up. They're in alphabetical order, so you should be able to anticipate this. And we should be able to get a lot of the remaining iClickers registered today. And then we can do it again next Monday until we finally get this done. Okay, any other general questions? All right. So we'll start as we've been doing. So here are the 2 questions. The first one is from the last lecture. Has to do with concordance and discordance. As usual, probably we'll have a little discussion on this one, but choose the best answer. I got the grape with the caffeine, so I'm ready to go. All right. Should be able to go to 1 minute on this one. Okay. 3, 2, 1. All right. What's the answer? All right. Okay. So most were in agreement this time. So the idea here is it's arguing that there's not a strong genetic component to this. Okay. And then one that should be simple for today's lecture, if you paid any attention at all or prepared it all ahead of time. And about 75% of you didn't. Okay. All right. We'll go to 1 minute on this one also. No, now it's 50-50. Yeah, that's about what I expected. We can go back to chapter 3, probability. Okay. 3, 2, 1. There we go. So what's the answer? No. Oh. Oh. So when you're a neurosurgeon, you're not going to get to go, oh my God, my hand slipped. Now he's brain dead.
It's alright, I won't be practicing in the US.
Oh, cool.
Yeah.
Neither do I. Okay. So the answer to the second question, it was in Drosophila. It came out of Morgan's lab. We'll come back to it. So we're going to finish chapter 6, which is really human genetics. It's a fairly short chapter, and a big part of that is for you to begin to see a little bit about some of the limitations in terms of ethical, practical, technological limitations to work doing human genetics, but at least talking a little bit about what we can do with these. And that's really where we ended up last time, was really in the realm of genetic counseling. You have a couple, they want to have a baby, but there's some concern about some genetics that they might have inherited from their parents. One is we can test ahead of time. Now, in the past, it was a lot of it was just a pedigree analysis, very much like what we'd seen before. Is there a history in the family? It was an uncle. We can do some probabilities. For most of these diseases now, at least the really well characterized ones, we can just simply test the individuals directly. But then the next step, and I believe this is pretty much where we, let's see, is this where we, so I think the slides before this, the next step was, okay, you have a couple who are each heterozygotes for some recessive genetic disease, cystic fibrosis. So there's a 25% probability that their child would have the disease, but they still want to have a child of their own biological origin. So then you can do in vitro fertilization, allow the cells to begin to divide in vitro in the lab, and after, in the very early stages, around the 8, 16 cell stage, this very early little blastula stage, remove one cell, and then you can do a full DNA analysis using the polymerase chain reaction and some of the other technology we'll talk about later on. And you can test the individual embryos to see whether or not they're homozygous recessive for that trait. If they are, then you can select another one. So you can do pre-select the embryos prior to implantation. The other technique was, okay, once the pregnancy is underway, how do we begin to do different types of analyses? Ultrasound will give us some idea if there's gross deformities, but its resolution is not really that good, although it's getting better. Then there was amniocentesis and the CVS, where you're actually looking at getting a sample of the fetal cells, doing karyotyping, DNA analysis, and such. These are both very invasive techniques. So what's looking much more promising is this ability to really do a genetic analysis on the fetus just from a sample of the mother's blood. And it's the idea that unlike it turns out that actually a small percentage of the fetal cells do in fact cross the placental blood barrier, and they are in the mother's circulating cells. The challenge was, how do we pick out this 1 in 1,000, 1 in 10,000 cells? And there's a technique called the FACS, for fluorescence activated cell sorter. Dr. Hartman has one of these instruments. Without going into the details of it, it literally can sort cells cell by cell and can sort of kick off and say, okay, these cells are of fetal origin, these cells come from the mothers. And then you can do a DNA analysis on the fetus just to confirm that you really are looking at fetal cells. Then you can compare the father's DNA because they're going to be essentially hybrids. And so this is a very promising type of less invasive way to do genetic analysis of the fetus. We're not going to go into any of the techniques now, but you've probably read about it. I mean, some people are even attempting in utero modifications and such. Oh, this is what I was just saying about the pre-implantation. Okay. Postnatally, there's a number of genetic diseases that newborns have been tested for decades. One of these is PKU. We talked about this before. This is a pleiotropic gene in that it affects several different phenotypes. It's really involved in metabolism, the proper metabolism of phenylalanine. If it's not properly metabolized, there's a side product that accumulates that's very toxic to the nervous system. So at least during development, this can lead to mental retardation and some other abnormalities and deformities. And really, by testing early, you're able to intervene, modify the diet. Then the child develops normally. Once they reach adulthood, then they can have a normal diet. So this metabolism problem with phenylalanine is only during development, or only during the childhood. Okay. This actually goes back to genetic testing, and this is where we'd also talked about, why do these alleles of these genes, if they lead to such deleterious diseases, why do they persist in the population? And the short answer is they're hiding out with the heterozygotes. So the heterozygotes carry these alleles, but because they're recessive traits, at least at the clinical level, the individuals are unaware that they're carrying these traits. And then there's, if both parents are heterozygotes, then 25% of the children will develop the disease. With certain diseases, The alleles that lead to these devastating diseases are present at fairly high concentrations within certain populations. One that you probably know about is Tay-Sachs disease, at a high population in certain Eastern European Jewish communities. And here, because of the allele frequency being so high, the chances of two unrelated individuals both being heterozygotes is much higher. It's a devastating disease. These children rarely survive through their teen years. But now you could screen the parents ahead of time and identify the ones that are the heterozygotes. So for many of these, the incidence of these types of devastating diseases should drop dramatically by making use of this technology. Okay, so I think some of this is we talked about last time. This is another one that now starts to get involved in a little bit of foretelling of the future. So it's one thing to be a heterozygote and a carrier for a recessive disease, a disease which is recessive in nature because it's not affecting your life directly. Huntington's disease, and again, you may already know this, is an autosomal dominant mutation. So the gene responsible for this is a gain-of-function mutation, but it's a late onset disease. It typically doesn't show itself until probably around age 40 or so. So for you, that's off in the future. For me, that's, I don't know, somewhere back in the rear window somewhere. Okay. So you know there's someone in your family has had Huntington's disease or something that was diagnosed as Huntington's disease. And now, if you and your partner want to have a baby, what's the probability that that child will have Huntington's disease? Okay, so it's a 50% probability that you may have the allele, or a very high percentage probability. And since it's a dominant mutation, the probability, now the danger to the child is much higher. But what about you? You know, let's say you're thinking about having children and you're the very young age of 25, okay? Which for me was too young to have children, but that's a different point. But let's say you're 25. But now, do you want to know this? No, it depends on the person. Because this is the penetrance of this disease is virtually 100%. At this point, it is a devastating neurological disorder that is fatal after a period of years. There is no treatment for it at this time. And you can either roll the dice and find out that you get a clean bill of health and that this sort of Damocles is not hanging over your head any longer, or you may find that you in fact do have that allele and this now profoundly changes how you look to your future. I've read, I'm not a clinical person, but I've read that for this type of genetic analysis, the law actually requires that a psychologist be present when talking to the patients because it is really foreshadowing their future. Okay? But that's when we get to the point of what the genes can tell us about predispositions and such. This is where the technology is going, and there are going to be personal and social outcomes that have to be dealt with. Okay, so while you think about that, let's just try this question. Yes. Yeah, that's what I meant. Yeah. That any type of a maternal blood test. No, because you really don't know if you, for in vitro fertilization, you're going to have a sperm sample with a million plus sperm. There might. The question would be, how would you test a single sperm without destroying it? Oh, then if you're going to do that, you would just, if you do that, then you just test the patient ahead of time. Right. Yeah. If the man carries the gene with those sheer numbers, the probabilities are still the same. No, those. Um, the question is, how do you test the eggs without destroying the eggs? So, with at least once the embryo, what you really have is a clonal population where you have a discrete number of cells that are still totipotent. Therefore, you can safely remove one cell, analyze it, and still have it intact. These other approaches would all destroy the sample. So you'd answer the question, but you couldn't go anywhere with it. Okay. So what's the obvious answer? Okay. Oops. Just a couple of other areas. I'm sorry, what? You want to see the answer? What is the least invasive? We good? Okay. Okay. So here comes into the question of evolution. To my understanding, Florida hasn't outlawed it yet. They're probably working on it. Okay. But if you look at the science, you look at the data, since full genomes have now been sequenced, if you look at the genome of chimpanzee and you compare it to the genome of humans, it's about 95 to 90%, 95 to 96% homologous. So what I want to do in this slide is not argue the point of how similar chimpanzees are to humans genetically, but look at what's different. What's in that 5% that's different? And so I just think this is a really kind of an interesting idea in terms of how potentially, this isn't proven, this is just some evidence for an idea where small genetic mutations can eventually have profound effects in the divergence of species that were originally closely related and now they're divergent. So for humans, one of those major advancements was speech, the ability to clearly communicate with each other, develop language, written language, the ability to transmit information from one generation to the next. Well, there's a gene in humans called FOXP2. It's been shown to be required in human speech, and there are several speech pathologies for humans where it's been linked to mutations within this gene. If you look at it, this is one of those genes that's really quite different between chimpanzees and humans. And so if you look at it compared to mice and humans and primates and such, it looks like it's probably this mutation, this divergence in the sequences of the FOXP2 gene has been fairly recent, all right, suggesting that it may go back only about 200,000 years. That's about the time when we think modern humans started to emerge. So it just leads into this tantalizing idea that maybe these very simple genetic mutations that now set the foundation for something as complex as speech really become that touchstone that then allows the evolution of that species to go off in another direction. Whether this really happened, I don't know. I think it's just a really interesting idea. So this is what's coming out now as more and more genetic analysis is being done, more genomes are being sequenced, larger numbers of organisms are being found. There was recently a discovery of there were two women anthropologists found sort of a new species. It sort of seems to be this transition between primates to humans. I haven't been able to read the whole paper yet. But as we keep filling in these gaps, a little bit clearer picture of this continuum is starting to develop. So I just use this as one example of, instead of asking why are the sequences so similar, but maybe the interesting points are what about the sequences that are different? So to kind of just wrap this up, what's going to end up happening as genetic analysis becomes more detailed. I personally think we're very close to the point where newborns will probably be run through a whole panel of, let's see, there's about 25,000 genes in the human genome. Pick the 5,000 genes which are most frequently involved in some sort of human health condition. Screen these individuals early on. We may be able to start to get to see some predetermination. In other words, maybe a certain combination of alleles would suggest that this individual is going to be more susceptible for diabetes. Start changing lifestyle early on. It could have some very, very positive impacts. On the other hand, could it be used against you? There's already been cases of basically genetic discrimination by insurance companies. One case I read about, a woman was suspected that her daughter might have a congenital, basically inherited a genetic mutation. She had her tested when she was 10 or 12 years old, and unfortunately it was positive. The insurance company found out about this information. This was before Obamacare came into play. I don't know how it would be affected now. And they basically said, we'll insure the child until she's 21, and then we're going to drop her. Okay, and the mother had to sue in court as a case of genetic discrimination. Now, we do know that since there's a push to put all the medical records into databases, that all of your information will be completely private and won't be available to the insurance companies because these are unhackable systems and no one will ever get this information. Right? Raise your hand if you actually believe that. Because I got some pixie dust up here too that I can sell you. All right. So here's what's going to happen is this information's out there. The genie is out of the bottle. The question is, how do we as a society use this information? So even if you're never a professional scientist, you're never a professional healthcare provider, you just end up going into some other more lucrative field or whatever, you still have to have this basic understanding of genetics because it impacts your day-to-day life so much. Okay? So here's where I think this is what genetic counseling in the future is going to look like. I'll be able to tell you the sex of the child. It's a boy. He's going to be a smoker. He's going to be gay. And worse than that, he's going to be an insurance agent. Okay. And he has a 75% probability of developing Alzheimer's. Okay. So that's just kind of a very broad overview of what we can and can't do with human genetics at this time. Any questions on this? Otherwise, we're going to get into the next chapter, which is much more scientific meat to it, and we've got to get into it a little further. Okay. So this is chapter 7. This is on genetic recombination and linkage, and how this leads to gene mapping. So you probably sort of remember back to general biology, the big A's and the little b, the big A's and the small A's and the big B's, and they get switched around. You got red chromosomes and blue chromosomes and such. Does that make sense? There was some chapter on that or some slide or something like that. Okay. Now we're going to go into detail about how that very important process comes about. So this is going to be a second mechanism that's a driving force in evolution in terms of giving new genetic variation to each generation. What we're going to be doing here is putting different alleles of genes, or putting the alleles of different genes into different combinations with each other. So first of all, before I even get started into this, we are always going to have to be talking about at least 2 genes. Genetic recombination makes no sense if we only talk about 1 gene. Okay? If you may remember, I think it was all the way back in chapter 2 or 3, there was a diagram with a gene. It was just showing the segregation of the chromosomes. It did show crossing over, and the fact that you really couldn't detect that crossing over had happened, and that's because there was only 1 gene. So we're always going to, in order to be able to detect that this happens, we're going to be always talking about at least 2 genes. Ultimately, where we're going with this, and we probably aren't going to get there today or get close to it, is the so-called 3-point cross, or using 3 genes to do genetic mapping. So with that as an introduction, so here's the answer to that second question, was Drosophila. So Alleles genetics is really coming out of Morgan's lab. He's the leader. and using Drosophila as a genetic model. This is the early part of the 1900s. And so Alfred Studevant was another student of Morgan's. And as they started identifying more phenotypes and the associated genes, what they found was lots of examples of pairs of genes that did not assort independently, as Mendel had said. So there were now numerous so-called apparent violations of Mendel's laws. And it was really Morgan who was the first to suggest that what they were really looking at were genes that are on the same chromosome. So as I talked about before, back when we did chapter 3, Mendel was, for nothing else, very lucky in that he looked at 7 traits. And if I remember correctly, pea plants have 7 pairs of autosomes, and that he was essentially looking at traits that were encoded by genes that were on different chromosomes. Okay. Now there is one particular dihybrid cross that he should have done, but he didn't do. It's not in his paper, and had he reported it, he would have seen the gene linkage that we're talking about here. So the question is, did he just not happen to do that cross because he didn't have the plants? Or did he do those experiments and got results that were so different than everything else he was looking at, he just put those aside because they were too confusing? But that was 150 years ago, so we'll let it go. So what we really start looking at is the opposite of what Mendel had. Mendel was looking at independent assortment, that the probability of inheriting a particular allele that affected the seed shape had no influence on the probability of inheriting the allele that would affect the color of the seed. What we're looking at here is just the opposite. Traits that are being inherited together, and that's because they physically reside on the same chromosome. So this is the breakdown of the chapter. So the first part is we're going to compare inheritance of linked genes, which are those that assort independently. So independent assortment was what Mendel saw. Now we're going to look at, well, what's the different situation? The second is, and we might begin to get to this point today, is how genetic recombination breaks up these gene linkages. So this is this variation that's being put into place by putting different combinations of alleles together. We'll talk about that, how that was used to construct gene maps. And the third, which we won't be able to get to today, are what are some of the methods that we can use to identify the physical position on the chromosome. So maybe it's worth mentioning right now that if we do this gene mapping and such, as we go through all of this, what it really tells us is the relationship of one gene to another, how far away it is, and whether it's on the same chromosome, but it cannot tell us the position on the chromosome by itself. We need other data to be able to do that. Okay. So this is just a review of Mendel's principles, which everybody's completely familiar with because the exam is on Wednesday. So he really gave us these 2 major ideas. The principle of segregation, that is, he correctly deduced that diploid organisms, which are the vast majority of organisms on the planet, at least eukaryotic cells, have 2 alleles of each gene, and that those alleles segregate, one allele segregates into each gamete. And then the idea of the principle of independent assortment, when he did his dihybrid crosses, they found that the segregation of alleles for one locus did not influence the segregation of the allele for the other locus. And that's because he was looking at genes on different chromosomes. So we can kind of look at this in a diagrammatic way. And so now we're going to introduce a new idea. We're going to have non-recombinant and recombinant gametes. And this is going to be a term that's going to come up a lot through this discussion. So this is a Mendelian cross. Okay, does this look like a why is this a Mendelian cross? All right, so they're homozygous. They're homozygous for homozygous dominant here, homozygous recessive here. So this is a typical dihybrid cross that Mendel would have done. You only get one type of gamete. All right. You have fertilization, and the F1 is now the double heterozygote. Okay, all that looks familiar because this is what you've been studying, right? Yeah, they're all going, yeah, okay. Now we go to the F1 generation. Now we can get two types of gametes can come out. You can get big A, big B, and small b. I'm sorry, actually 4. These two are what we're going to call the non-recombinant. That means they're the same gametes that you would get from the parents. There's been no recombination. Big A is still with big B, and small a is still with small b. So that's the non-recombinant gametes. The recombinants mean we've recombined it. We've created a new combination. Now we have gametes big A, small b, and small a, big B. They're different than the parents, and so these are going to be the recombinant gametes. Right? The secret to what we're going to be doing here is when this frequency of recombinant gametes is 50%, you're probably looking at independent assortment as Mendel saw. If this number is significantly less than 50%, you're looking at genetic recombination between two genes that are on the same chromosome. Yes? I don't think so. All right. What we're going to be really looking at is how often do we get these recombinant gametes? In Mendel's case, it was 50%. So it's 50% non-recombinant, 50% recombinant because they're independently assorting, and it's equally likely that you get one or the other. So if you are looking at a problem and you see this kind of cross and you get 50% recombinant, you're probably looking at unlinked genes. But if it's significantly less, 20%, 10%, 30%, then you're looking at genetic recombination that's between two genes that are on the same chromosome. Okay. So that's what I just said here. So 50% of the gametes will have the recombinant gametes if those two genes are on different chromosomes. Okay, and this simply is the result of what happens in meiosis. The homologous chromosomes can align independently along that metaphase plate, and so they just segregate independently, and if you look at a large enough number, 50% of the time you're going to get recombinant gametes, 50% non-recombinant. So that's everything that we've been looking at up to this point. Genes that are physically close to each other on the same chromosome are said to be linked. The old genetic jargon, which is still around, is called a linkage group. So if these two genes show recombination significantly below 50%, they're in the same linkage group. Physically, that's telling you these two genes are on the same chromosome. And that these linked, and you can see this because when the genes are linked, they tend to be inherited together. Okay. So here's the question. Do the genes for flower color and pollen shape in sweet peas assort independently? This is one of those things that Mendel was, this is the kind of question he was asking. All right. So we start out, we got purple pollen, we have purple flowers along pollen, those are the dominant phenotypes, red flowers, round pollen, the recessive. Do the fertilization, you get the F1. Does this look like complete dominance, codominance, or incomplete dominance? Yeah, this is a Mendelian type complete dominance that we're looking at. Okay, so now you're going to have self-fertilization, so it's really the heterozygotes are going to now cross with each other. You go into the F2, and you find you get 284 with the two dominant phenotypes, 21 that have one dominant phenotype and one recessive, a similar number that now have red flowers, long pollen, and 55 that have red flowers and round pollen. Okay. Does that look like a Mendelian cross? Well, maybe sort of. Okay. you expected that 9 to 3 to 3 to 1. That's where you would see it, that 9 to 3 to 3 to 1 ratio, if you had independent assortment. But you're not seeing that. So something else must be going on. So right here, you do not find that expected 9 to 3 to 3 to 1 ratio. And what you're probably looking at is gene linkage. So what's happening is these two genes are on the same chromosome. And the key here is that during meiosis, so remember we talked about in meiosis, you have crossing over. So, but that doesn't occur in every single cell, it just occurs occasionally. Now, a very important idea of where we're going with this is, the closer the two genes are to each other on the chromosome, intuitively, would that increase or decrease the number of recombinant gametes you would see? It would decrease it. Overall, there's some exceptions, but in general, the crossing over is a random event. As long as it occurs somewhere between those two genes, you will see the recombinant gamete. So just statistically, the closer the genes, the less likely it's going to happen. The further apart the genes, the more likely it's going to happen. So we're going to use that frequency as a way to eventually build up some gene maps. Now here's a very important part, because we don't want to make this too easy. You can have genes far enough on the same, apart on the same chromosome that their recombination frequency approaches 50%. So they appear to be unlinked. So if that recombination frequency is getting up around 50%, you're probably looking at unlinked genes, but you have to keep in mind it's possible that you're looking at two genes that are very widely separated on the same chromosome. This was clearly the case for two of the genes that Mendel was looking at. So they would appear to independently assort. But we'll talk a little later, we'll talk about how to deal with that. Okay, so this is really just going to show, going back and reviewing meiosis, to show you where these chromosomes, when this happens. So here is that 4-strand stage, the tetrads. So this is the two, you got the pairs of homologous chromosomes, this is meiosis. They're now aligned so closely together, you actually have 4 chromatids, but they're all aligned with each other, and crossing over can occur, and it usually then shows up as these little chimeric chromosomes. So this blue chromosome has now exchanged some information with the red chromosome, and that's when recombination has occurred. Okay, so the genes may switch here, homologs, which is literally all they're doing. They're really switching one gene from one homolog to the next. All right, you go through meiosis 2, and so in the end, you actually end up with 4 one-end cells. Are any two of these cells genetically identical? Would they be genetically identical if no crossing over occurred? 2 of them would be identical, right? Okay. As far as we can tell, a successful meiosis requires at least 1 crossover for every chromosome. So it's possible that occasionally you may have a meiotic cycle where no crossing over occurred, but that's very unlikely. Is this the only crossover event that happened? You just get 1 per chromosome. No, we're just looking at 1 so that we can even think about this. Okay? So you can have, the same event can happen multiple times along the chromosome. Okay, so the key here is genes, alleles that had previously been linked, the big A and the big B allele were on one homolog, small a, small b on the other, have now, their linkages have been changed. Now big A is linked to small b, and big B is linked to small a. That's all that genetic recombination is really doing. Okay. So let's see what you can do with this. All right. So with these questions, you can collaborate with each other. So you have a heterozygous parent is test crossed to a homozygous recessive, what will the progeny phenotypic ratio be if genes A and B are completely linked? So by completely linked, I mean they're so close together that crossing over between those two genes has never been observed. Well, the answers are all over the place. I mean, this is probably a reasonable approximation of a question you'll see on exam too. I'll go to 1 minute and 30 seconds on this one. All right. 3, 2, 1. All right. So what's the answer? Well, the crowd was quiet. All right. Okay. So we can learn a lot from this one. So why is it C? Those two genes are so close together that recombination never separates them. Would these be, so are these recombinant or non-recombinant gametes? Right, because you got big A, big B, which you got from one parent, and small b, small a, small b from the other. So they're simply traveling together. They're on the same chromosome and they're so close together that the probability of recombination occurring between them is too low to be picked up experimentally. What would the answer be if these two genes were on different chromosomes? Then it'd be B. That's what Mendel saw. So these are the two extremes. Either the genes independently assort because they're on different chromosomes, or they never separate because they're linked so closely together physically on the same chromosome. And then the third is the reality that, yeah, they're on the same chromosome, but they're far enough apart that once in a while recombination occurs. How often that happens will give us an indication of how far apart those genes are on the chromosome. Okay? Yes?
Recombination doesn't just to clarify the term, the crossing over creates recombinant gametes, but regular independent assortment also creates recombinant gametes. But would you also say recombination occurs?
Well, when we're dealing with recombination, we really can only deal with two genes. So independent assortment is going on. Crossing over is occurring all along this chromosome. We're just focusing our attention on these two genes.
But the recombination doesn't only apply to crossing over, it also applies to this non-crossing over when you have recombinations of gametes.
Yes, but if the gene,
example?
right, if the genes are on different chromosomes, then for those genes, then you're going to have the non-recombinant gametes. So it's going to depend on, I think an example I got a little bit later, when we deal with two different chromosomes, I think you'll see, we just have to think about, even if we're dealing with 4 genes, we have to think about them in pairs. Okay. Any other? So here's a little bit of the nomenclature. Was there a question or just stretching? Oh, okay. So here's an important point. If the genes are linked, that is, they're on the same chromosome, then it's also important that we know which allele is linked to the other allele. So if you think about it a little bit, you got 2 genes, they're on the same chromosome, they're heterozygotes. There's 2 possible orientations for the organization of those genes, and that's what we have to be able to indicate in our nomenclature as we go through these genetic crosses. So this is the way we would indicate a double heterozygote for a Mendelian cross. There's no markings other than we're just showing big A, big B, small b. So if there's no other indications, this is telling you that these genes are unlinked. Okay, this is what we've been looking at. If they are linked, then there's two possible configurations of the alleles, as I just said. So a common way of doing that is to use a line to separate them, and above the line are the alleles for one chromosome, for one homolog, and then below the line are the alleles for the other homolog. So we could either have big A paired with big B on one chromosome, and small a with small b on the other homolog, or the converse. We can have big A, small b on one chromosome, small a, big B on the other one. Now, when we get into these recombination experiments and the mapping and such, all that's going to happen is, if this is what it is to begin with, this is what recombination gives you. If it starts here, recombination gives you this. So you just have to know what the starting pattern is. Then you'll know when you see the recombination, it'll be the opposite pattern. Now, this is a little bit clumsy to draw. So many times in a shorthand, what you'll see is a slash. So you can put all this on one line, but it means the same thing. This is the same as this, and this is this one. Okay. So what we're looking at here is the opposite of independent assortment. All right. So as we just talked about, is a situation where the genes were so close together, recombination just doesn't happen. The target's too small. Think of it that way. If recombination is at random, the important point is that crossover, that recombination can occur anywhere between the two genes. If it occurs outside of the two genes, then it happened, but it's not going to show up in our experiments. All right. So here's an example. This is going to compare the two extremes, either completely linked or independent assortment, which is what Mendel saw. So we've got this plant. We're dealing with two genes, just labeled M and D. One is mottled leaves, and the other has to do with the height. Okay. So let's do the independent assortment over here. So is this a test cross? Sure. You have an individual who's wild type for both phenotypes, so you don't really know what their genotype is. Crossed to a parent that has both recessive phenotypes, so it must be homozygous recessive for both genes. So this is a Mendelian, this is a test cross. All right. We can get 4 different types of gametes out of here. All right. We can get big M with, what do we got? The 2 large alleles, the 2 small alleles. Is that the recombinant or non-recombinant? Well, one is it doesn't, okay, take my word for it, okay. It doesn't really matter here, right? Well, I guess, no, technically it would. So if this is independent assortment, what gamete, you're getting gametes, I'm sorry, I'm getting ahead of myself. So you got big M, big D, got so many, small m, small d. So you got the 4 different possible gametes, all in equal proportions, just as Mendel would have seen in a dihybrid cross. Just one type of gamete comes out of the test cross, right? So now you fertilize, you do the cross fertilization, and you find that you get equal numbers. It's usually sharper than this. All right. So you get the non-recombinant progeny. What are they? They match the parents. So this has the genotype of this parent. This one has the genotype of this parent. Okay, over here we have the recombinant progeny. These two, the phenotypes here, do they either one match the parent? No, the genotype doesn't either. So here are the recombinant progeny. All right. The key here is this is 50% are non-recombinant. So that tells us the 2 genes are linked or unlinked. If you get 50% recombinant, then it's unlinked because they're independently assorted. So this is a Mendelian cross. Here is the other extreme. Here the genes are linked together. So immediately what it should tell you is there's only going to be two phenotypes that come out of the progeny. They're going to have to be non-recombinants. So here we have to diagram the allele configuration because they're linked. So what this is trying to show you is one homolog has the two dominant alleles, one homolog has the two recessive alleles. This is the test cross, so both homologs have the two recessive alleles. All right. When we go to gamete formation, if there is no recombination, if these two genes are so close together that there's no recombination, how many different types of gametes can you get? Just the two. All right. And then you get the one type here. When you do the crosses, you restore the genotype of the one parent and the genotype of the other parent. So you get all non-recombinant progeny. So when you never find the recombinant progeny, that's telling you these two genes are not undergoing recombination. They're so close that the chances of recombination occurring between them are too small to really be picked up experimentally. Okay, so these are the two extremes. Independent assortment, which is what Mendel saw, and complete linkage, meaning there's no recombination between the two genes. It means they physically reside very, very close to each other on the same chromosome. Do we know which chromosome? No. Do we know anywhere, are they near the telomere, are they near the centromere? Tells us nothing about that. It just tells us about the relationship between those two genes, their relative positions on a chromosome. Okay. Confused yet? I can do better. Okay. I'm trying. Actually, I'm trying to be clear with you. You got to follow this through. Okay. So we know crossing over occurs in prophase 1 of meiosis, and it's really a genetic exchange between the non-sister chromatids. Would you normally have genetic recombination between sister chromatids? No, it shouldn't happen. It's usually when it does happen, it's an indication of some kind of DNA damage has happened. If it happens, can you detect it genetically? Yeah, so if you have recombination between sister chromatids, detecting it would be theoretically impossible. If they're genetically identical, they've had genetic exchanges, but they're still the same. What you're seeing, you're picking it up, you can see it here because you got non-homologous chromosomes. Okay? If the two parents are homozygous for the same allele of a gene, for the same allele for each of two genes, could you see genetic recombination? No. You have to have differences. You have to be able to see some sort of a difference. That's why most of these experiments are all going to involve a double heterozygote. Okay. Here's an important point. You got 4 chromatids that are paired together at prophase and meiosis. Only 2 participate in the recombination event, and 2 remain unchanged. All right. So this is a very important point here. Only 2 of the 4 chromatids are involved in the recombination event. 2 remain unchanged. So I'm leading up to something that's going to be very confusing, so stay with me. Until you go, yeah, now I see it. So the chromatids that undergo recombination have new combinations of alleles. So let's look at this more in a cartoon form. So here's the 4 chromatids at meiosis in prophase. So we have the red, the blue. We have the sister chromatids, so they have the same alleles. We go through meiosis 2, and at the end of meiosis 2, we have 4 gametes, that are each now one end. And has any recombination occurred? No, they're exactly the same proportion as before. Now, let's assume recombination did occur, and that's what this little, we usually call this crossing over, so that's what this little X is trying to indicate. So what's really going to happen is you come along this chromosome, down here, you're going to drop down here and over here. So now red big A is going to be linked to small blue B. The converse, and this is an important point, under normal conditions, if nothing's going wrong, these are reciprocal events. That is, each chromosome gains and loses the same amount of information. So the reciprocal crossover would be blue A coming up, crossing over onto the red chromosome, and now red B. But what happened to this chromatid and this chromatid? Nothing. So at the end of meiosis 2, this chromosome and this chromosome are now recombinant. They now have the alleles in new configurations. Here, big A was linked to big B, and small a was linked to small b. Now, we have put them in new combinations. Big A is linked to small b, and big B is linked to small a. These are the two recombinant chromosomes. But these two remain non-recombinant. So only 2 of the 4 chromatids participate in this recombination event. With me so far? Okay. So for each meiosis, a single crossover gives you 2 recombinant and 2 non-recombinant gametes. So this really, at this point, is no different than what Mendel saw. 50% recombinant. Okay, keep that thought in mind. So it's exactly the same as independent assortment. If this crossover occurred in every single meiotic cell, we would have 50% recombinance, and we couldn't determine whether these genes were linked or unlinked. The difference is, the closer the 2 genes are together, the greater percentage of meiotic cells in which the recombination does not occur. So what we're really looking at and asking is, what percent of the meiotic cells underwent this recombination event? This is the part that's confusing until you think it through. One crossover generates 2 recombinant gametes. I just showed you that, right? And 2 non-recombinant. Therefore, the percentage of recombinant gametes is half the percent of meiosis that underwent the recombination. And that is why the maximum recombination frequency is 50%. This point will come up over and over again. So let me try in this diagram to see if, because that, does that seem intuitively correct? Or it's confusing? So just walk it through this example. Assume you have 50 meiotic cells. So they're going to produce 200 gametes. We agree on that? Okay. Assume 2 recombinant gametes. Out of this whole thing, we're going to, and then, so what's going to happen is in 49 of those 50 cells, there's going to be no crossing over. We go through meiosis 2, and they're all, these are recombinant or non-recombinant? These are all non-recombinant. In one meiotic cell, out of those 50, we have 1 crossover. And out of that, we got 2 recombinant gametes. So we said we're producing 200 gametes, and we got 2 that were recombinant. So we have 1% recombinance. What percent of the meiotic cells underwent recombination? 1 out of 50, or 2%. Because 2 of these chromatids weren't involved. So the recombination frequency is half the percentage of meiotic cells that underwent recombination. Okay? Should I do that again? I don't know if I can do it again, but I'll try. All right, we're going to start with 50 meiotic cells. Everybody in agreement we'll get 200 gametes out of that. Okay. Let's assume that out of those 200 gametes, 2 are going to be recombinants. So that's a 1% recombination frequency. Okay? 1% of our gametes are recombinant. To get that, 49 cells undergo no recombination. So these are all non-recombinants. The only recombination, so out of these 50 cells, the crossover between these two genes only happened one time in one meiotic cycle. So this meiotic cell is going through meiosis. This is prophase 1, and there's crossing over. But the important point is, and the key to all this, only 2 of these 4 chromatids are involved. So the crossover occurs here. So we get 2 recombinant gametes, but 2 remain as they were. So out of these 200 gametes, only 2 are recombinant. That's a 1% recombination frequency, right? But what percent of the meiotic cells underwent recombination? 2%, 1 out of 50. So the recombination frequency is half the percentage of meiotic cells that underwent the recombination. And that's all we're really measuring, is what percentage, when we do all this gene mapping and everything else that we're going to be doing, it's just asking what percentage of the meiotic cells underwent recombination.
Could you just repeat the summary, please?
Okay, so we're agreed we have 200 gametes.
Just the summary here of the actual rule.
Okay, oh, okay. So what this means, since only 2 of the 4 chromatids are involved, the percent recombination will be half the percent of meiotic cells that actually underwent the recombination. In this case, we have 1% recombination because it occurred in 2% of the meiotic cells. If we had a 10% recombination, what percentage of the meiotic cells?
5.
10, 20.
20. Okay. So if 10% recombination means 20% of the meiotic cells underwent recombination. The other way to think about it, what's the maximum recombination frequency? 50, and that would happen if recombination occurred in 100% of the cells. Okay? Does that make it easier to see? So 50% is the maximum number of meiotic cells is 100%.
So basically you said with the recombination frequency is half the percent of meiotic cells actually undergo.
Yes. Okay, so let's see if you can use that information to answer this question. So for single crossovers, the frequency of recombinant gametes is half the frequency of crossing over because of what I just said. But that's not one of the choices. Okay. Now there is an answer up there that a lot of you are probably going to put down. Just because it's correct doesn't mean it's the best answer to the question. One is an outcome, the other is asking why. I'll go to a minute and 15 seconds on this one. Most of them are in. Good, and it looks like most Most of you are getting it right. Okay, 3, 2, 1. Okay. So what's the answer? Yes. So it's just because only 2 of the 4 chromatids are actually involved in the crossover. Okay. Any questions? All right. What do we got here? Oh, now we're going to do some math. Almost here, the whole class just gets nervous. Math is, believe it or not, math actually is your friend. Okay. So we're going to calculate recombination frequencies. So we're going to be dealing now with linked genes. So we go back to this plant, whatever it is that we're using. These 2 genes, M and D, are they linked or unlinked from the way this is drawn? They are linked. So they're both on the same chromosome. All we're going to do is try to calculate what % of meiotic cells undergo a recombination between these 2 genes. So keep in mind, it doesn't matter whether the crossover occurs very close to M, very close to D, in the middle, or anywhere between them. As long as it occurs between the 2 of them, the outcome is the same. Okay, so we can get gamete formation. Which of these gametes would be the ones that there's no recombination? These 2 over here, right? So they're in the same configuration. Big M is linked to big D, small m is linked to small d, just like they were in the parent. So these are the non-recombinants. Okay. Now we can also undergo crossing over. So let's assume we have crossing over. If that happens, why do we still have non-recombinant gametes? Because those are the 2 chromatids that weren't involved in the crossing over. And here are the other 2. All right. So, okay. So these are the 4 that you get out when you have crossing over. How many genotypes from the test cross? Just the 1. Okay. So now we have fertilization. All right. One of the first things you want to sort of as you build up a thinking about how to deal with these problems is, if we're dealing with linked genes, the most abundant okay, first of all, I said these crossing over is a equal event. The 2 chromosomes should exchange equivalent amounts of information. So these gametes should appear in approximately the same proportions. So we have 55 and 53. So you look at the progeny, and the first thing to do is just look for the phenotypes or the genotypes, whatever information you're given, and put them together in close numbers. They should occur at there should be 2 of them that are almost the same frequency. So you got 55, 53, that makes sense. Put those together, and 8 and 7. Very important point. The most abundant group will be the non-recombinants. Okay, the recombination is going to be rarer. So if you're trying to figure out, are these the recombinants or are these the recombinants? Well, there's a lot more in this phenotype. So from there we deduce. How do we know? Okay, so all we really can look at are these plants. These are the 4 genotypes, 4 phenotypes that came out from the progeny. But how do we know that this is the genotype? Right. In each of these, throughout this whole diagram, it's showing you the 2 homologous chromosomes. Do we really care about this lower chromosome? We only need that to convince us that, yes, this plant has both dominant traits. We know it has the 2 recessive alleles from the test cross parent, so it must have both dominant alleles. But once we know the genotype, don't let these confuse you. That lower chromosome is coming from the test parent. These are really, this upper chromosome, those are the 4 gametes that came from the from the heterozygous parent. All right? And so what this is going to really tell you is this is the configuration of the alleles as they were originally in the parent. Because these are the non-recombinants because they're the most abundant. The rarer ones are the recombinant progeny. Okay. Now to determine recombination frequency, all we're really going to do is ask, how often do we get recombinant progeny? So if you just add them up, we had 8 plus 7, so we had 15 recombinant progeny out of a total of 123 that we looked at. We just add them all up, 55, 53, 8, and 7. 15 divided by 123 gives you 12.2%. So you have 12.2% recombination. All right, let's round it off to 12%. Okay. So now let's test your short-term memory problem. Same problem, same answers. If need be, maybe I will give you credit for E if you just throw up your hands in desperation. Okay. The recombination frequency is 12%. What percent of meiotic cells had a single crossover between the M and the D gene? So far, only one person's been honest enough to answer E. I'll go to 1 minute and 15 seconds on this one. So 3, 2, 1. All right. So what's the answer? Okay, just what we said before. 12% recombination frequency means 24% of the time in that meiotic cell, as it was going through meiosis 1, crossover occurred between these two genes. Okay. So I'll give you a little more background on this, and then in a few minutes from now I'll stop and we'll answer questions, which usually means everybody leaves. But I gave you the opportunity. So for linked genes, the arrangement of the alleles on the homologous chromosome is critical. Because what you have to do is, the key to solving these problems is first figure out what the allele configuration is in the parents so that you can recognize the recombinant situation. And you're going to see that because we're going to talk about two orientations, cis and trans. Well, if they start out in cis, recombination just puts them in trans. If they start out in trans, recombination puts them in cis. So you have to figure out what the configuration of the alleles are in the parent. If you don't do that correctly, everything else falls apart. All right. So we have a double heterozygote. Most of these problems is what we're going to have is a double heterozygote cross to a test cross. So we'll do a fly. This is a fly example. So we have a purple thorax is recessive to the normal green. In this case, it's with flies. So the P, again, the designation is typically assigned to the mutant phenotype. So this mutation of the P gene gives you a purple thorax. The plus indicates a wild type allele. Same thing here with the puparium. That's the little pupal case where the larval undergoes metamorphosis and goes from a crawling eating machine to a flying sex machine. Okay? Flies have a very simple life. It's also short. Then you got the color of the puparium. Black is recessive to the normal brown color. Okay. So here's what's really important. The configuration of these two genes. So we're always going to be talking about two genes when we talk about a specific recombination frequency. If both wild-type alleles are on one chromosome and the recessive alleles are on the other, they're said to be in the coupling or cis configuration. The opposite would be one wild-type. The wild-type alleles are actually on different chromosomes. So you got one wild-type and one recessive allele on one chromosome, and then the wild-type and recessive on the other. This is repulsion or entrance, so basically pushing against each other. So you either have the two wild-type alleles together, they're in cis, or they're on opposite chromosomes, they're in trans. So here's the key point. Again, and what I'll do is I'll take you through this one example, and it'll be a good point, I think, to come back to when we regain some sort of, not sure what I want to say at that point. When we come together again next Monday after the exam is behind us. Okay. In a test cross. So we're going to have a test cross of a double heterozygote. The first thing you want to look at is that predominant group of progeny. That's going to tell you whether those alleles were in cis or trans in the mother. All right. Now, I haven't really gotten into it at this point, but with flies, there's kind of a quirk with flies. Genetic recombination does not occur in males. This is not in general flies. It may occur in other flies, but in Drosophila, genetic recombination does not occur in males. So when we set up these fly crosses, the double heterozygote will always be the female. The test cross will be the male. So here's the test cross, homozygous recessive for both genes. So you just get the one gamete that comes out of there. Here are the not. So this is in the. So here's an important point. What's it? If you got, in one case the alleles are in cis or coupled, the other in trans or repulsion. What about, can you tell that from the phenotype of the mother? Does the mother have different phenotypes? No, she's a double heterozygote, so she has the wild type phenotype for both genes, right? So we don't know starting out whether in this female, this double heterozygote, whether those genes are in cis or trans. That's what this experiment is going to show, and also show you that the outcome is exactly the same. It doesn't matter whether they start in cis or in trans, the outcome is the same. All right, so here, we're just given that they're in cis. So does everybody, assuming you can see this, does everybody agree that these are in cis? Okay? So you're only going to get the one gamete from the test cross male. So here's what we want to focus on, are these four gametes. Here are the two non-recombinants. One is just this upper chromosome, both wild-type alleles, and the other has both recessive alleles. Here are the recombinant gametes. All right, do you see how this went from cis to really trans? So now you have one wild-type and one recessive allele in each case. All right, so now you put these crosses together. Here is the four phenotypes. You're going to get four phenotypes that come out of this. We can deduce what that genotype is because we know that one homolog has both recessive alleles. Why do we know that? Where did that come from? That came from the test cross father. So that's what this lower chromosome is, all the way across here. This lower chromosome, we don't even worry about it. The upper chromosome is really showing you what the gametes are. These are the gametes that were in the egg from the mother. All right. So here's the two wild-type alleles and the two recessive alleles. Do they occur in about equal numbers? There you got 40, almost two perfect, 40 and 40. Okay. Over here, you have two phenotypes which are in similar numbers, but only about 10. So we know this represents the original configuration in the mother. So in this mother, the two genes were in the cis configuration. All right? So you got 10% recombinant progeny. Over here, we'll flip this around and we'll let the mother start in the trans configuration. Same thing, we have the non-recombinant chromosomes, except now each of these has one wild type allele, one recessive allele, right? And then over here, the recombinants have two wild type alleles and two recessive alleles. Put them all together, ignore this lower chromosome, which is coming from the test cross male. Now, which group is the larger group? Over here with 40 and 40. So this has got to be the configuration of the alleles unrecombined in the mother. And they're in trans. Here are the recombinants, which are now in cis. Did the outcome change? You still have 20% recombinants. So you get 20% recombination between these genes, regardless of whether you start with a heterozygote who's in the cis configuration or the double heterozygote who's in the trans configuration. So if it starts out in trans, recombination puts it in cis. If it starts in cis, recombination puts it in trans. Okay. All right, why don't we stop with that one? I'll leave some time now to do two things. One, let me shut this down so I don't lose the files.